Hey everyone, these are your notes for membrane structure and function. This is going to go on the back of the mega sheet that you guys have for cell biology. So on the back side we have notes about endosymbiosis, cell size, shape, pro versus eukaryotes. So I'm taking up an entire half square on the back of that mega sheet for the notes on membrane structure and function. So one of the big and common recurring themes for our class is how structure affects function. And so in this case, what we're going to be looking at are the specific components of that plasma membrane and then how that imparts structure onto the cell itself and the importance of it. Now, in ninth grade, you guys learned that the plasma membrane or the cell membrane is what is known as semi-permeable. And part of the reason that it's semi-permeable is because of its structure. So when I ask you for the components, when I ask you for the structure that makes up the plasma membrane, we're going to be looking at specific macromolecules, and we're going to be doing them kind of in order of importance. So the first one we're going to look at are the lipids. There's two types of lipids involved in the component or the structure of the plasma membrane. The first of which is the bulk or what makes up the plasma membrane itself, and those are phospholipids. Okay, so those phospholipids have a hydrophilic head and hydrophobic tails. And the head is the polar or hydrophilic area, and the tails are the hydrophobic or the non-polar areas. The second type of lipid involved in the plasma membrane is cholesterol. And the cholesterol is used to maintain fluidity. So it's involved with how fluid a membrane is going to be. So in addition to the unsaturated tails, you can also have cholesterol embedded within that phospholipid bilayer to help increase or decrease fluidity depending on which direction you are heading. The second type of macromolecule that is important or a component of the membrane are proteins. There are two types of proteins that we are going to discuss. We have transmembrane proteins. These are the ones that are going to span across the membrane itself, and we're going to see these act as channels. The second type of protein that is a component of the membrane are what are known as peripheral proteins. And these only stay as one side. They can act as anchors. They can act as part of the third compound, which are carbohydrates and identification. So let's go ahead and write that down. So your carbohydrate chains are going to be used for identification. And if it's a protein and a carbohydrate together, we call them glyco proteins. So glyco comes from carbohydrate. Proteins are proteins. If it is a carbohydrate and a fat together on the plasma membrane, we call those glycolipids. So glyco, again, carbohydrates, lipids are lipids. What we're going to do now is we're going to draw the membrane structure. And I'm going to draw that over here. I'm going to incorporate these components into our semi-permeable membrane. And then we're going to discuss how that structure affects the function of the cell itself. So what I'm going to do is I'm going to draw several phospholipid heads. And I'm just doing them in blue to remind you that they are hydrophilic. I'm going to draw several tails. And I'm going to do these in red. And it's important that you understand that the tails themselves in this bilayer will form a hydrophobic interior of the cell. So this area right here is hydrophobic. And it's going to repel because the interior is non-polar, it's going to repel anything that is polar. So I'm going to add up here to our definition. So we know that the plasma membrane is semi-permeable. We're also going to call it a bilayer. And the whole reason it is semi-permeable is because of those hydrophobic tails. I'll say that again to emphasize this. The whole semi-permeable nature is dependent on those hydrophobic tails, those non-polar tails repelling against polar substances. So anything that has a charge is considered polar. And so anything that comes along that has a charge is not going to be able to cross through the plasma membrane. We also have transmembrane proteins that act as channels for substances that do need to cross a cell but otherwise wouldn't be able to. So a good example of this is aquaporin. Is a specific channel for water to pass down its concentration gradient and into the cell. Remember that water is polar, so water would normally not be able to pass through this hydrophobic area of the cell or of the membrane to get into the cell. So specialized channels, these transmembrane channels, are going to allow for things, for substances such as water, anions, cations, to go down their concentration gradient. The amino acids of the protein channels, specifically the R groups that are interacting with the hydrophobic area, those will be hydrophobic R groups. The R groups that are interacting with the aqueous environment, either inside the cell or outside the cell, those are going to be hydrophilic R groups. So remember that with the amino acids, the carboxyl, the central carbon, and the amine group are the same, but the R group is different, and the R group can be hydrophobic or hydrophilic, and then change the way that the protein folds, either in the primary or the secondary, the tertiary, and the quaternary level of protein folding. I'm going to draw a few more phospholipids. Again, this is in a bilayer. We have the hydrophobic tails facing interior, the inside of the cell. Some of these are saturated, some of these are unsaturated. We could also have cholesterol to help maintain the fluidity. And so I'm just going to draw a cholesterol molecule right here, just as a generic shape, nothing too specific. Remember that cholesterol, since it is non-polar, since it is a lipid, will be embedded within the tails themselves. We also have peripheral proteins, so peripheral proteins will stay on one side of the membrane. So here is my peripheral protein. Okay, it'll either be inside or outside. And the outside of the plasma membrane is going to have different structures than the inside of the plasma membrane. So out here in the extracellular environment, the channels, the proteins, the carbohydrate chains will be different than the ones on the inside of the channel. And these peripheral proteins can move along the surface of either surface of the inside or the outside of the plasma membrane, similar to the way that a boat would move on a lake. Remember that your membrane does need to be fluid. It does need to have a certain consistency to allow these protein channels to move, to allow the shifting and changing of your plasma membrane, and so that cholesterol will help to maintain the correct, what we call fluidity. It should be flexible, and if you were to feel a plasma membrane, it should feel something similar to olive oil. Lastly, your carbohydrate chains, and carbohydrate chains are just sticking out. Good example of these carbohydrate chains would be blood typing. And so if you have A blood, if you have B blood, AB blood, or O blood, it's carbohydrate chains on the exterior of that plasma membrane that allows for that. So to recap, the macromolecules that make up the plasma membrane, or what we call the components of the plasma membrane, contribute to the structure and the function of that plasma membrane. The phospholipid tails, being hydrophobic or nonpolar, are going to control what substances can come in and out of the cell, and to substances that are nonpolar, such as oxygen, such as carbon dioxide, would be able to diffuse down their concentration gradient into the cell, whereas polar substances such as water, cations, anions would need a specialized channel. They would need to be facilitated with these protein channels, these transmembrane protein channels, to allow them to move down their concentration gradient in the absence of energy. And so it's important that you guys know the structure of the plasma membrane, and it's also important that you guys know how that structure imparts function in maintaining substances inside and out of the cell. Thanks for watching.